Good afternoon. This is Victoria Berry presenting the video lecture connected with the histology of the endocrine system and how to label the histology slides. Thankfully, I happen to have Office 365 with my home system, which has some very handy visual labeling features. So I'm going to speak about the technical side of that first, just how to label. This is my home computer, and in the label menu, up at the top, you just use in the insert menu. And in the insert menu, you have the option of shapes. One of the possible shapes that can be used is a straight line with an arrow. So whatever structure you're attempting to label, you just place the arrow wherever that structure is. Then for the labeling portion of this, what I use is actually a text box, which is also in the same menu. You just drop down and you select the option where it says draw the text box, and it will pull up a cursor that is shaped like a cross basically, and you just drag it across and it creates a box. And at that point you just type in the name of whatever the structure is, for example, like nucleus of the pituitite. So you just type it in there, and then save it in that way. And in Office 365, thankfully that works very, very well. Okay, so moving on, I'm going to speak about the actual glands which we reviewed in these histology slides. So the first one was the pituitary gland, which is actually very interesting that everything with the glandular system begins deep inside of the brain. The pituitary works in conjunction with the hypothalamus, both of which are structures deep in the brain, and they create pretty much a domino effect of messages to other endocrine glands to signal them to secrete their hormones. The two types of cells that we were looking at, it's kind of obvious in the histology slide that we have here in our lab report. The upper portion of it has a lacy structure that really looks more like neurological tissue, and the lower half of the slide is sort of clearly more of a lobular structure. So the nucleus those are the small dots that are within this lacy kind of tissue. Then the other two cells that were in this slide specimen are the acidophil, and those can be distinguished somewhat. They stain a little bit lighter in the what would actually be a microscope slide. A basophil is a little bit darker, so that was about the only way I could really distinguish between the two. Then we move into the thyroid gland, which is very profoundly important to the metabolism of the body. And we see follicular cells and colloid. Colloid means fat, and actually that's pretty obvious in this specimen by the big circular structure that is the colloid. The epithelial cells are in almost single file along the edge of the colloid. Moving on from that, we have the adrenal glands. This has a much more complicated structure. where it's in multiple zones, all of which, I'm just trying to get this on the screen. Okay. Um, so we have, they're called zona glomerulosa, um, which is to the interior. So in the middle of the slide here, the capsule, which is like the perimeter there. There's also something called the zona fasciculata, which is a portion here. You can tell there's a slight change in the color there, and the zona reticularis. Then moving on, we come to the pancreas, which is very critically important and can malfunction in diabetes. And we have the islets of Langerhans. The islets and the acinar cells are both critical relative to insulin production and the release of glycogen from the liver. The ovary sample, which is the next endocrine gland in our lab report, was quite easy to identify because the follicles were of a good size. So they can be seen here just as a circular structure that is to the center of our slide. Okay, and the last specimen is from the male testes, and it shows the spermatogonium, which is the developing sperm. The only difference between that and the spermatocyte, it's really tough to distinguish in the slide that we have, but one is more formed than the other. The spermatocyte is more formed, where it's a more clearly circular structure as compared to the spermatogonium, which is the developing sperm cell. And the very last structure that we have is the Leydig cells, which produce androgens in the male, which would be testosterone and related hormones. And I thank you very much for your attention to this lab video.